Hey guys, welcome to our last chapter in this class. Chapter 20 we have divided into 4 topics. In this first topic we're going to talk about the extracellular matrix and connective tissues. So here's our topic outline. We're going to talk through what is an ECM, what are tissues, how are plant tissues formed, and look at how animal tissues are formed. As always, here's your topic objectives. Please let me know if you have any questions on these so we can talk about them in class. But these are what I expect you to have mastered before our exam. Alright, so all semester we've been talking about individual cells. We rarely talk about cells when they come together. But as you should hopefully have put together by now, the cells are really important because they come together and form tissues for multicellular organisms such as you and I. How does this happen? Well, this is through a variety of junctions and connective tissues and structures that help bring these cells together to form the tissues that we're talking about. One of the main components of this is the ECM, which is the extracellular matrix. And it's defined as a complex network of polysaccharides, proteins, and other components that are secreted by the cell. And it's kind of like the glue that holds things together. And this is what gives us the support in tissues such as bone or wood. It gives us our strength, it gives us our flexibility in our skin, and it's a way that holds the cells together. And you can see how there's a variety of cells within that, and we're going to talk about that in topic 3. Right now we're just talking about how the cells come together. So what is a connective tissue? A connective tissue is defined as an organized mass of cells with a specific function. So this can be anything within the body as long as it has a specific function. So your skin, your gut, etc. And so you can see here how there's a bunch of connective tissues and how all these cells have come together to form different structures to perform different functions. And that's what's important about this, is tissues are very specific to what they're supposed to be doing. And plants and animals evolve differently in how these connective tissues are built, so we're going to talk about them separately, but it's important to know that they are different types of tissue, or that the tissues are built in different ways. So let's start with how plants form tissues. Now, we haven't talked about plant cells hardly at all this semester, but plant cells have a strong cell wall. Remember, we talked about that briefly, that they have this cell wall. And that makes it so that their ECM has to form very differently than ours because of the fact that these cells on their own are already fairly structured and fairly rigid. And what they use to build their ECM and thus their tissues is cellulose. And cellulose, as you can see here, are these big green rods, and they're running through the primary cell wall, and then eventually will be moved into a secondary cell wall. And this is what provides that structural support. And really the cell wall is just another type of ECM, and we'll talk about ECMs here in a second again when we look at the animal tissues, but it's just a space between the cells. And it's very, very important that you understand that the cell wall is specialized and it mirrors the function of the cell. If the cell is supposed to be a very strong, structured cell, the cell wall is going to be like that. If it's more of a flexible or a waxy cell that's on the leaf, the cell wall is going to reflect that. Another aspect of this is almost everything we've talked about that the cells produce is produced internally and then exported to the surface of the cell. In this case, this is not the case with the cell wall. The cell wall is synthesized on the outside of the cell, so it's synthesized in place, which makes it pretty unique. Alright, so now let's talk about animal tissues. There's 4 types of connective tissue. Or 4 types of animal tissue: connective, epithelial, nervous, and muscular. And connective is really the only one that's different from the other 3. The other 3 all have similar tissue structure designs. So we're going to look at connective tissue over the next couple slides. But it's important that you understand that there's 4 types. We're just going to look at connective for the moment. And they can range in all sorts of toughness and flexibility, from being very flexible like your skin to being really tough and dense like your bones. It just depends on what we need that tissue to be. And the bulk of the space between these cells in your tissue, or in your connective tissues, is filled up by the ECM. So it's important to understand that. Alright, so let's now look at how the animal cells form tissues. In plants we saw they used cellulose. In animal cells, we use collagen, and collagen is the main component, and it actually is a main component of any mammal. About 25% of all the protein in any mammal at any given time is collagen, and it's a stiff rope-like helix then is twisted again to create these collagen fibers that run throughout the connective tissue. And this is created within the cell and exported to the ECM, unlike what we just talked about with the cell wall, where the cell wall is created on the outside of the cell. This is created inside in parts and is exported and then formed together there. And each cell within these networks have a very specific function, and they're very specialized. So fibroblasts work on the collagen network to help make it the structure that we know that it is, or that we need it to be for each specific type of tissue. So each tissue will have these collagen fibers running in their own unique way throughout the tissue. So how do the cells connect to this network of collagen fibers? Well, they don't connect directly. They can't. The connection is just not that strong between them. So what we have is a series of proteins that connect through this process. So we have the collagen fibril, and what attaches to this is something called the fibronectin. The fibronectin is a protein that is found in the ECM, and it will connect to the collagen fibril. Then we have these integrin dimers, and these integrin dimers are proteins that are transmembrane proteins in the cell. And you can see that here. Now we know that the plasma membrane is not a very strong structure, so it's really important these integrins, after they span the membrane, are connected to something inside the cell. And you can see that here, where the integrin on the internal side of the cell is connected to actin through an adapter protein. And so this structure helps hold these cells in place and helps form this tissue. Now some cells can use this mechanism as a way to travel by connecting and disconnecting through that, and that's how they actually move around in tissue, such as macrophages or things like that. But this is also used to help keep cells stationary. As I mentioned, most of the space in the ECM is just there, and it's not, or most of the space in tissue is the ECM. And it's just there, it's not filled up with cells. And so what we need to fill in the gaps is this cushioning, and it's a gel. There's gels and proteins that help do that. There is a protein called the so you have the two macromolecules. We have the proteoglycans and the glycosaminoglycans, which is GAG. And you can see those here on this slide. And they form this kind of brush bottle look. And what this does is it allows for the cushioning of the tissues so that they can take a little bit of an impact without getting too much damage. The more firm we need the tissue to be, the more protoglycans are going to be, because that's the protein component. If we want it to be more jelly-like, like your eyeball for instance, your tissue is going to have a higher amount of GAG in it. And this is a careful balance, and it once again goes back to the specific nature of each tissue. So that's our introduction to ECM and animal tissues. If you have any questions, please let me know.